Great. Good evening, everyone, and welcome to APA New England's final speaker series webinar for 2026, New Sights into Food Allergies from Bed to Benchside and Homestead. My name is Jan Hansen, and I'm president of the Asthma and Allergy Foundation of America New England chapter. We're known as APA New England, and we're truly thrilled that you've joined us tonight for this special speaker series webinar. We know food allergies impact an estimated 33 million people in the United States, and they're affected not only with their physical health, but social and emotional quality of life. Why food allergies occur and why their prevalence is increasing in modern times, and particularly concentrated in urban areas, is not completely understood. And Dr. Andrew Wang, our expert speaker for tonight's webinar, will address the latest research that his group is pursuing in answering fundamental questions surrounding the underlying mechanisms driving food allergies. Dr. Wang, and he's asked, I can call him Andrew, so I'm going to do that. Andrew is a principal investigator at the Food Allergy Science Initiative, also known as FOSSI, and an assistant professor in the departments of immunobiology and internal medicine at the Yale University School of Medicine. Hold on one second. I would like to share just a little bit more with you about Andrew and his background. Andrew is a physician scientist who is an expert in inflammation and allergy biology and physiology. Clinically, he is an expert in sepsis and autoimmune and autoinflammatory diseases, and he's been performing research in immunology since high school. After receiving his AB with honors from Harvard University, he became engaged in extensive training. He completed his clinical training in 2015 and then pursued postdoctoral training in the laboratory of Dr. Ruslan Medzatov. Upon completion of his research training in 2017, Dr. Wang made fundamental discoveries in how the body anticipates and defends against environmental threats, including those posed by the modern environment, such as exposure to industrial chemicals and increased psychological stress. For his pioneering approach to tackling fundamental problems in biology and medicine, he was awarded an NIH Clinical Investigator Award in 2016. He was named as a 2020 Pew Scholar and inducted into the American Society of Clinical Investigation in 2023. Needless to say, we are delighted that Andrew is joining us tonight to share his information with us. Now, before we begin our program, for those of you who have joined us who may not be familiar with APA New England, we're a patient education and advocacy nonprofit organization, and our mission is to improve the quality of life for all those who have or are impacted by asthma, allergies, including food allergies, and related diseases. As a regional nonprofit, we serve all 6 New England states, and we've been doing this since 1979. We fulfill our mission for a wide range of activities, including our awareness initiatives, our legislative advocacies for needed improved public policies, our support for research for improved quality of life, our support services for our community, and our educational programming, such as our speaker series webinar this evening. APA New England is proud to serve our New England community and delighted that you're all here, as I've said. Tonight's webinar will be recorded and will be available for future viewing on our website at asthmaandallergies.org. And I'm going to ask for that to go into the chat for you, so that if you want to see this again, because there's going to be a lot of information shared, exciting information, if you want to see it again or share it with others, go to our website, asthmaandallergies.org. I do have some brief notes to share before we get started. We ask that all program attendees remain on mute for the duration of the program so that everyone will have the best possible audio experience. But if you do have a question during the program at any time, please feel free to type it into the chat. And we will, between Andrew and myself and Caroline Rochford, who's helping out tonight, we will make sure that that question gets asked and answered. Now, any mention or reference of a company, an organization, or institution, or their products or services shouldn't be viewed as a recommendation or an endorsement by AFA New England. And last, information presented in this webinar should not be used as a substitute for professional medical care to diagnose or treat a specific illness. And now, for our important webinar tonight, new insights into food allergies from bed to Benchside and Homestead. I am incredibly pleased to introduce you to our expert speaker, Dr. Andrew Wang. Dr. Wang, it's my pleasure to turn this webinar over to you.
Jan, thanks for the very kind introduction. I'm really delighted and honored to be here. Thank you also for everything that AFA is doing as a healthcare provider that's and scientist. The work is really, really important, and we're very, very grateful for it. I do have a couple of disclosures. None of them will relate to what I'll present to you today, but so that we're all kosher here. I want to start off first by actually thanking FASI. Jan mentioned them in her introduction. FASI is a relatively new, essentially consortia of a lot of really interesting scientists across the world at this point. We have folks over in Germany now that are part of the consortia who are doing work across the whole body because it's very clear that food allergy is not really just a problem of the gut or the immune system, but really affects probably every organ and how they communicate with one another. And the main interests of FASI include how food allergies get established in the first place, the allergic response and how that is actually triggered, because it's not, as I think as you'll see from the presentation I'm about to give, not totally clear. And then trying to understand why some people, although they may be allergic, have no response to food, while others have fatal responses. And the fundamental idea here is that's shared amongst all the scientists, is that we're trying to understand how the thing works, so that by doing that, we can come up with new and effective remedies. So the program for tonight, there are a series of events, vignettes of work that we either recently published or are about to publish. I'll start with a couple of patients, go over very briefly some of the factoids about food allergy that Jan already very nicely covered. Talk a little bit about what we know about food as a science, and then talk about some of the work that my group has been doing at Yale, which include our work on the skin, what's in modern food, and then this area that's very exciting to us right now about how the brain and the nerves interact with the immune system in our food. So we'll start with a couple patients. The first one is a 25-year-old female with joint pain and a butterfly rash. And she was diagnosed with lupus, and this is my mom. And this is why I got into doing research as a high school student, because I was going to doctor's visits with her, and it was quite frustrating. Second patient is a 6-year-old boy who developed an anaphylactic response at a Pick Your Own just down the street here in Guilford, Connecticut. And this is my son, my oldest son, who's, this is a younger picture of him with many of you may recognize this. This is called the sign of Denny Morgan. He's quite atopic. And so this is the reason I ended up directing a lot of my laboratory into studying allergic inflammation. And I think the question that my group started to tackle first was why this is happening so much more now. And this kind of segues into the epidemiology of allergy. So just to point out that it's not just food allergy, which is, and allergic diseases, which is on the right. This is from my good friend at University of Virginia, Sir Tom Platts-Mills. And he points out essentially that this is a modern disease. And this, you can see that it arises really at the turn of the century. And different diseases like asthma and allergy start to arise only kind of in modern times. On the left include autoimmune diseases, right? So many of you may recognize multiple sclerosis, type 1 diabetes. where the immune system is, instead of being targeted towards bacteria and viruses like they're designed to do, get targeted to your own tissue. And autoimmunity and allergy have a lot of things in common. The immune system has no business attacking stuff that's not bad, right? So in one case, it's food. You shouldn't attack it. In the other case, it's your body. And as you can see, the incidence of these disorders also really exponentially increases after the 1950s, very similar to allergic diseases. If you zoom in, so if you zoom out and just ask, on the left is rheumatoid arthritis, another autoimmune disease. On the right is food allergy. And the darker the color is the more prevalent the disease. So rheumatoid arthritis on the left, food allergy on the right. You can see that it's actually kind of a non-random distribution. And in general, these are in industrialized countries. You could argue, well, this could be genetics. You know, the folks in Western Europe are genetically very different than the folks in India and China and so forth. And so a particularly illustrative example, and there are many of these. At this point, there are some that published out of our colleagues at Northwestern within the United States. If you zoom into single countries and even single states, in our case here, the increase in the prevalence is really exclusively in urban areas. So when you look within states, or in this case within countries, and you look at where the prevalence of, in this case, rheumatoid arthritis, but the same is true with food allergy, where is it increasing? It's increasing only in industrial urban environments. And so, you know, these are kind of things you notice at practicing medicine also. I mean, I've been doing this for a little bit. When I started my lab, this is now eight years ago or so, we decided actually to take a big step back and just ask, in general, how much is known about food, right? Because if you want to understand food allergy, you probably should understand something about food, the science of food. And it turns out that we're still really kind of in the phenomenology age of food. So this was just published in the New England Journal entitled Diets. And really our understanding of food right now is fairly limited to observation, right? That folks in the Mediterranean eating Mediterranean diets have much less incidence of various diseases. This is, I think, and I get this a lot in the clinic. You know, doc, what can I eat that's non-inflammatory, or what's a less inflammatory or anti-inflammatory diet? And I think the fact of the matter is we just don't know. But certainly people are starting to think about this. This may remind some of you of a periodic table of trying to actually understand the composition of food and how it interacts with our bodies and our physiology. So I think I don't need to point out, I don't think, although this was a revelation for us, and I'll get to this in a little bit, in how we live, how my family lives now, this is what food was like when the vast majority of our evolution that made our genes the way they are. This was what food was then, right? And the main question that we had to address in this time when our genes were being honed to the environment was whether or not whatever it is that we found we could actually eat. Like, did it have nutritive properties? And more importantly, did it have properties that, although they were nutritive, could also poison and potentially kill us, and therefore compromise reproductive fitness in our genes and there, right? And so I'm putting up a couple of pictures here. I don't know if there's any mushroom foragers in the audience. This is a popular thing to do up here. Usually when I give this talk amongst clinicians, I'll ask them whether or not they can eat these various things. And so turns out if you eat this particular mushroom, and for the mushroom foragers in the audience, you may recognize this. This is the death cap mushroom, or Amanita. Every year in the ICU we will get, and in Germany this is much more common because there are a lot of mushroom aficionados there, will eat this. And this turns out to be something that is quite poisonous. The mushroom makes this poison to defend itself. And this is a general theme. So in the middle here, for the Agatha Christie aficionados, this is nightshade, or belladonna. And this is what it does to you if you were to ingest this. And on the very right, the Native Americans were using this here. This is the willow tree and the bark of the willow. They were using it for medicinal purposes, but if you notice some of the toxic properties the willow bark has, you'll notice that it is what your doctor tells you is the side effect of taking things like aspirin and ibuprofen, because this is where we got aspirin and ibuprofen, the salicylates, were extracted from the bark of the willow tree. And obviously the plants are making this for their own purposes to prevent herbivores from eating them. And many of these things now we have used medicinally to be able to solve various problems, like if your heart goes too slow. And amanitin, for example, is being developed for cancer. And so the fact is that the food makes a lot of things that defend itself, and so we have evolved many ways of dealing with that. And the point of this slide is just to impress you with all the different types of enzymes encoded by genes that we have that break down all of these toxic things, like aspirin. And if you reconceptualize drugs in this historical context and in the context of how we had to evolve with our food, it shouldn't be surprising that things like aspirin engage evolutionarily honed genes that we have to detect them and detoxify them as ways that we have evolved to be able to eat food and be able to survive it. And so this is work that we published a couple of years ago, but we made this very interesting finding that some of the NSAIDs, which have now been engineered for particular purposes, engage these poison defense pathways and some don't. And it probably explains why many of the NSAIDs, although they target the same thing, have very different effects in patients who take them, because some of them have different properties, and they're related to these evolutionarily conserved properties of detoxifying naturally occurring chemicals, which NSAIDs are based on. All right, so how do you survive this, right? So if you were to taste this, and usually these things are bitter, and they have very unique tastes, and so they engage the nervous system, our sensory system. And then if we experience something aversive to it, like we vomit, which is usually this response when we ingest some toxin, or we have diarrhea, or we feel nauseated or malaised, we associate this experience then with these sensory cues. So the next time, for example, you see this same willow tree, you probably won't eat it again, right? So this is associative learning. And it's really very similar to Pavlovian conditioning, right? So in this case, if the willow tree has this toxin in it, and the willow tree happens to taste like spearmint is obviously not toxic, but you have this very bad response to it, then you learn that these two things go together. So the next time you have experienced the taste of spearmint, for example, even if there's no toxin in there, you could still have all of the neurological memories that are associated with that very bad event that was very memorable. And I'm sure all of us have anecdotes about this. If we've ever helped a good friend have a little bit too much to drink, or other similar types of events with shellfish or other things. And immune memory turns out to be very, very similar. And so I'm going to spend a couple of minutes here, because this will be a very important concept to remember as we go through the rest of the discussion. Immune memory works in a very, very similar way. Obviously, this is a hugely blown up version of a bacteria. Obviously, you can't see the thing. It has no taste. It has no smell. And so how do you detect it? How do you remember this? But clearly, the immune system can remember it. So how does that work? So just like with neurological memory, it relies on contextualization. So in this example, so here I'm showing a very kind of pared down version of this, where let's say you eat a food like egg. Normally, the default mechanism, if you see that, is that your immune system says, well, there's nothing bad about this. This would be like spearmint flavor without the toxin. And we kind of remember it as being fine. There's nothing to remember, really. On the other hand, if we want our immune system to remember it, we have to put in, generally speaking, an adjuvant. And with in the case of vaccines, this is putting in adjuvants that make then something that's inert, like the spike protein itself, although it may have some toxic properties, without an adjuvant is not a particularly good thing for the immune system to remember. And so the adjuvant, which generally speaking all have inflammatory or aversive qualities, then contextualize the antigen into something that then we remember. So this is exactly the same as Pavlovian conditioning for neurologic memory. And so the aversive element here in immunology is called an adjuvant. So if you normally get infected with the bacteria or virus, it has all of it, right? So it has the spike protein, and then it has all the other stuff that has adjuvant properties. And so this is why when you get infected, you remember it. Okay, so you can see here that one can then conceptualize food allergies basically very similar to associative learning in the nervous system. So that the food itself has no intrinsic bad qualities, but if it's paired with something that's aversive, then the next time you encounter the food, you remember it as being something aversive. Okay, so with these kind of foundational things in mind, I want to tell you the first story that we published this year about the connection between skin and how we experience food. I'm happy actually to stop here to take any questions if there are any before I move on. If not, I'm happy to keep going. All right. So another one of our FOSSI investigators last year really beautifully discovered and identified all of the cellular components that allow us to normally tolerate food, and identified the cells that respond differently than when the food is, for example, coupled with something like a worm. This has been an outstanding issue, I would say, in food allergy biology, which is if the normal default path, based on everything I've told you about how the immune system remembers things, in particular with food proteins, is that it tolerates it, right? Because if you have to absorb nutrients to live, and so the default position of the immune system in the GI tract is to tolerate food antigens. How do you even get food allergy? And so for a long time, the field has converged on this idea of what's commonly called as epicutaneous sensitization or dual exposure, which is basically that that in the lung and in the skin, and I think the skin is probably the best studied, if there are breaches in the skin, and this is most commonly the form of eczema, that the antigen, in this case, in this cartoon, peanut, enters through the damaged skin. And in this way, since it's not going through the gut, that association between the peanut protein and aversion and danger of the damaged skin then together form immune memory to the peanut, which presumably entered through the skin. And similar arguments have been made in the lung. The skin argument particularly bothered me because, as Jen may have mentioned, I have 4 kids. My youngest is 2. And generally when their skin has issues, they don't like to rub food or peanut butter or any of these other things. They kind of baby it. And one main reason why this idea came about was in every case, and the vaccine is exemplary of this, the adjuvant and the antigen are given in the same anatomic space. So it's injected in the muscle together. When you get a virus, right, the antigen and the adjuvant are together. And so it had not been considered that it was possible that you could put the antigen and the adjuvant in separate places. And so this was basically what we sought out to ask, which is it possible that if you had skin damage and when your skin was damaged, you ate something that you hadn't eaten before, that the immune system would associate the damage in the skin with something dangerous about this protein that you were ingesting. And it could be what we ended up showing was that it could be anywhere. It could be under the skin somewhere else. It could be in the lung, and it could be eaten, and then form an immune memory to this. And so this is work that was published a few months ago, and this is done by an extraordinarily talented graduate student who's now at UCSF, and a student who's now an MD student at UPenn, and was co-supervised by my former postdoc, who's now a faculty member at Yale in the dermatology department. And basically what they did was they damaged the skin in mice in many, many different ways. So we abraded the skin by using tape stripping, which is the common way that scientists model eczema in mice. We also gave them blisters. We also just did a standard punch biopsy that the dermatologist, Anna, would do in her clinic. And we used also kind of synthetic models of atopic dermatitis with chemicals. And then we also gave them bad sunburns. And we asked the question, if the mouse experienced these skin events while they were given egg protein, would they then develop antibodies, which is the memory of the immune system to egg protein? And then would they have an anaphylactic response if we gave them egg protein back? And what you're seeing here on the right, the more yellow it is, the more antibody they have. And as everyone in this audience knows, IgE is the allergic antibody. And the short answer is yes. And if you give them then egg protein back in either the tape stripping setting or the punch biopsy setting or in the chemical setting, mice will have an anaphylactic response. And so here what I'm showing is we have a surrogate for blood pressure drop, which is temperature loss in the mouse. So they drop a lot of temperature very, very quickly because they're small critters. And this lines up pretty well with their blood pressure drop. And you can see basically not only do they make the IgE antibody to egg, but they also have an anaphylactic response to egg when they see it again later. And so the question was, well, how does this work? Because the egg protein is going in one place, the skin is way over here. How can those events be connected to an immune cell? And so Dan and Isabella and Anna considered, well, it could be something that is released at the site of injury from the skin that's now circulating everywhere and has access to everything, including the place where egg is absorbed. Or it could be neuronal. So the nervous system connects all the organs. And if, for example, pain is being broadcasted everywhere to the organism, it can coincide on neurotransmitters can be released and coincide on the same cell that also sensed this egg. The short version of this is that we looked at a lot of different things that are made into the blood after skin, and there was a common set of immune factors that were released from skin damage that we could detect in circulation at multiple times across multiple kinds of skin injury. And I don't want to get too bogged down in the details here, but what Dan could then do was test the ones that we could detect in the blood individually and show that just adding this, so now we're not doing any damage to the skin, we're just adding the thing that the skin releases when it's damaged, the protein, in a recombinant form. And we can essentially make, we can recapitulate the effects of the skin damage. So we could give just, for example, IL-33 back, and then with the egg either under the skin or in the mouth or in the lungs. And then in doing so, we could form immune memory to the egg. And then when we challenged them with egg, they would have an anaphylactic response. We ended up working out many of the details of how this works. I just want to show you a couple of things here. One is that if, and this is should be very reassuring. It was, at least to me with a food allergy child, that if there was early exposure to, in this case, egg, in the animal model, so when they had it right after birth, skin damage afterwards couldn't break that oral tolerance that was developed from early exposure. So this is for the for many of the people in the audience, they're probably familiar with the LEAP trial and how early exposure to peanuts is protective of developing food allergies later. And so the same thing is true here, so that further skin damage later, as long as the system has already decided that this protein is not dangerous, cannot break that previous tolerance. The other thing is that the system is very, very tunable. So a little bit of skin damage doesn't do it. It has to be significant skin damage. And here you can basically see the more of this IL-33 that we add with the egg protein, the more egg antibody we get. And as one would expect, the more anaphylactic the animal is as a function of the amount of IL-33, which here is a reductionist way of skin damage. So it's really a signal to noise, and the system is trying to decide, is this actually dangerous and should I remember it? Because the memory, as many of in the audience know, once it's formed, is very hard to forget. And so what we discovered here, and this is a very short cartoon summary of the discovery, is that in addition to this kind of local priming model of this epicutaneous sensitization where the peanut is getting into broken skin, there probably is also a remote priming system in place where there can be skin damage and then food that's not been seen before can be consumed in that context, and that could be remembered as something that is dangerous and we should form an allergic response to. I think this has wide-ranging applications, as one can imagine. Many of the adjuvants we use in vaccines now are very complicated mixtures of things. And what we've essentially demonstrated here, and I'm not going to go into all the details, is that you can actually just give one single protein with whatever it is, spike or flu or whatever it is you want to immunize against, and have very predictable responses, antibody responses. And so this has implications beyond just food allergy, but into immune memory in general against a wide variety of things. Okay, the second vignette I'm going to talk about is our work in trying to understand modern food and its relation to food allergy. And I just shown you that immune memory requires an adjuvant, and we talked about this from the conceptual perspective, and then I showed you an example in the skin. And this was the paradigm that we were talking about earlier, right? So that without an adjuvant, an antigen is not something that the immune system remembers. And for the longest time, and Tom was a champion of this, was the idea that because we are so much cleaner now after the hygiene, after sewage and antibiotics and so forth, that the immune system kind of had nothing to do, and so decided to kind of run amok and cause all these problems with now being directed at things that it should have no business being directed at. And the work I'm going to present is, again, work that Anna did this time as a postdoc in the lab, and then Brandon, who's now an MD PhD student at University of Colorado with the same team, Dan and Isabella. And they came up with the very interesting observation that at the same time we had hygiene, we also had essentially the chemical revolution, right? So Bayer introduced aspirin right at the turn of the century. And then in the 1940s, we needed to be able to extend the shelf life of modern food as part of the industrial food movement. And then in a big way, pharmaceutical companies tried to make salicylates into more designer type drugs. And these were also introduced from a historical perspective around the same time that the hygiene hypothesis would posit that it's the lack of immune activity that may be causing all these problems. And so what we thought was, if the previous example is can I eat this, the modern example is even more challenging, because now it has not only nature-made chemicals that plants use to defend themselves, but man-made chemicals, mostly for preservation purposes. Our approach to this was actually coming through the food additive list from the FDA. And I just want to give one example of some of the things that we found to be hard to understand. So this is what the FDA says can be added into boiler water, so water used to prepare foods. And you can see here that some things can be in steam contacting food except milk and milk products. There are things that can be in steam contacting food with no mention of milk and milk products. And this is quite a long list of things. And it occurred to us that it may not be entirely clear if we understood the biological properties at least as it related to how our bodies would see this, in the same way that our bodies would see poisons from plants. If any of these had toxic properties that had adjuvant properties. And so the hypothesis was very simple, which is prior to 1960, we had the same antigens that we have now. And for those of you who know, evolutionary timescale is not 50 years, it's like 5 million years. And so it's unlikely that peanuts, eggs, their genomes have evolved meaningfully in the last 50 years. But rather, as opposed to the hygiene hypothesis where things are taken away, this is kind of a hypothesis of something added. And the hypothesis was that maybe we added something into industrial food that had adjuvant properties, and that as a result of this, we are now having allergic sensitization to something that's otherwise not dangerous in itself. And so this is kind of memory by proxy, right? So the same idea of Pavlovian conditioning. What's being remembered is actually this, but since the immune system can only see proteins, this is now being used as a surrogate. And so the experiment was very, very simple. We, again, took egg protein, and then we screened a whole bunch of FDA compounds. And this is kind of a standard way that we induce food allergies in mice. Essentially, it's a prime and then a boost. And then we asked, do they make IgE antibodies to ova? And then can we anaphylax them to ova? And so one of the first things we tested were actually NSAIDs, because these are commonly things that we give young children around the time that they would be exposed to food. And for the clinicians in the audience, you'll know that there are a whole set of NSAID related clinical entities. For example, with NSAIDs causing nasal polyps and asthma and allergies to aspirin. And so what you can see here, and this is, I'd appreciate it for those on the call not to distribute this on social media because this is still in revision at Nature. We screened a whole bunch of these NSAIDs. So here's ketoprofen, ketorolac, ibuprofen, indomethacin, and so on. And we identified two of them, ibuprofen and indomethacin, that induced, that had adjuvant properties, that induced IgE responses to egg protein, and then caused animals to anaphylax when they were reintroduced to eggs. And this was work that we've been building here for some time now. And you remember I showed you the slide earlier that certain of these NSAIDs induce these detoxification defenses that were probably designed for naturally occurring poisons that we might accidentally eat while we were trying to find food. And it turns out that the only NSAIDs that were able to have adjuvant properties were those ones that were able to engage these poison detoxification mechanisms. So, for example, indomethacin and ibuprofen engage this detoxification mechanism, whereas ketoprofen and acetaminophen and ketorolac do not, and we screened a whole bunch of them. And those are also ones that do not engage this detoxification pathway. I have my colleague in GI has this posted outside of his office, which I find quite humorous. When Bayer initially advertised aspirin, they famously wrote that it does not affect the heart. And obviously, we know that if you think you're having a heart attack, you should choose some baby aspirin. All right. So then on to the food additives. So we went to the FDA list. We picked a whole bunch of them. We screened probably 100 of them or so. And at the concentrations that the FDA says that we can add into food, so this is usually weight by weight. And we picked ones that are commonly used in preserved food groups that are allergenic in this country. So we picked peanut butter, dairy, and egg. And then we had processed meat also in there. And then we mixed them all together like they would be found in food, added them to ova, egg protein, and then looked to see if there was anything in this whole bunch that had adjuvant properties, kind of as a batch screen. And you can see there's something in the peanut butter collection and in the dairy and egg collection that have adjuvant properties, but nothing in the processed meat one does. And so then Anna, Brandon, Isabella, and Dan then painstakingly tested each individual one from this initial batch. And I'll just point out one of them. We discovered several, actually. The one that's worth noting here is tert butyl hydroquinone, which is commonly used as a shelf life stabler for fats. And it's a very potent allergic adjuvant. So you can see animals will anaphylax, the mast cells will release their contents into the blood. And if you expose ova with TBHQ, it's sufficient to make the animal remember that ova is something that they should remember in a dangerous way. So this is a summary of essentially what we discovered. There are a set of these additives that have adjuvant properties. They all share one molecular feature, that they're electrophiles, and we worked out down to the molecule on the cell how this thing works in terms of forming immune memory. What I want to point out here conceptually is just that these are protective mechanisms designed to protect us. So the responses that are happening in the gut when these poisons are being sensed lead to very beneficial things like expelling the thing, vomiting, diarrhea, and sending signals that tell to the brain saying that, yeah, this is aversive, I feel malaise, I really shouldn't be eating this. And then our immune system remembers the protein always associated with the toxin, because then when we see this again, there's a very, very fast response that allows us to expunge this ingestion much, much faster than experiencing all of the toxic side effects that it may give us the first time when we don't have immune memories. This is exactly the same concept as vaccination or having previously experienced an illness. It protects you against the illness again because your immune system remembers it and can anticipate it. All right, and I'm on to the final vignette. I'm again happy to stop here with any questions if any come up. And if not, I'll keep going. And the last is our interest in the brain and what role the brain and the nervous system may have in food allergy. We were inspired by this observation that was made in the 19th century by a physician. It's famously called the rose cold. He had heard about a patient who was allergic to the sight of roses. And to test this, he presented them with cloth roses because he thought maybe it was the pollen or something. So in this particular case, it was truly totally inert because it was cloth. And the woman had an anaphylactic response to the sight of roses. And so the assumption that anaphylactic or allergic physiology can only occur upon exposure to the antigen, peanut protein, rose pollen, storage protein, is not, from a categorical perspective, not true, right? So you can generate anaphylactic responses with other sensory cues, not necessarily just exposure of the immune system to the protein. And we found this to be very, very related to the phenomenon that the brain has in most diseases of the immune system. So it is well known that acute stress makes most things worse. And so multiple sclerosis is a good example. Cancer is another good example. This is a very famous example of testing airway responsiveness in asthmatic children who were subjected to take, when they sampled them when they were taking midterms and finals or not. And the short version is that acute stress makes inflammation generally, including allergic inflammation, worse, which as a clinician never also sat quite well with me because as many of you know, when you're acutely stressed, you have this fight or flight response, and the adrenal glands make catecholamines and make cortisol. And we use these medicinally to suppress the immune system, right? So beta blockers, beta agonists, and prednisone, cortisone, are things that are probably our best immune suppressants. And so really what should happen when you're acutely stressed is that these diseases should get better, not get worse. And so a number of years ago, we made this very surprising finding that there are some immune signals, and we showed this in mice, but also in humans, that are actually part of the fundamental fight or flight response that had been overlooked. And so this turns out to be IL-6, which is it's a drug target. It's conventionally thought of as an immune signal. And so if you just, for example, switch cages in a mouse, so it's in a novel environment, it'll make IL-6. If you restrain it, like it would be, pinned down by a cat, it would make it. If you isolate it, so social isolation will induce it. And if you just actually bleed the mouse, it will, while it's awake, it will make it. And you can see basically the time scale of its release. So here is cortisol, here is catecholamines, and then IL-6 happens a little bit later. And so if you make the animal unconscious with ketamine or with halothane gas, this stress response of making this inflammatory mediator no longer occurs. And I should mention this is beautiful work done by Reina, who's a PhD student now at Yale, and my former postdoc Hua, who's now a lead scientist at Genentech. And so this is just to point out that there's still a lot about the fundamental biology of many things, including stress. And I would argue that more stress is certainly also a feature of our modern environment, have yet to be discovered. And the last story I want to tell you within this within our work on the brain is our recent discovery. This was published in the last few months, of the presence of immune cells within the brain tissue itself in just at steady state in mice and in men. And so this is beautiful work done by Tomomi, who is a graduate student. She's now at UCSF getting her postdoc, and a consummate neurologist scientist, David Hafler, who co-supervised Tomomi with me. And so for a long time, this is work dating back to the 1940s, the brain was thought to be a place where there could not be immune cells. And if you were to ask most people, normally there should be no cells in the brain. And as Jen might have mentioned, we homestead, so we grow a lot of the food that most of the food that we eat. And so here's my oldest daughter, no, sorry, my younger daughter, with a duck that we later ate, and here are some of our yearling lambs, and we do all the things that homesteaders do. And so because we kept chickens, and I'm curious about other animals, it was well known maybe 40 years ago that there were immune cells in certain parts of the brains in chickens, and chickens are vertebrates like us. And this was also observed in the rats, and this basically was never followed up. And so I'm going to show you a movie here now. We made a way of looking at all of the immune cells that are, and in this case, these are T cells. These are the cells that are important for making antibodies. Every white dot you're seeing is a T cell, and what we've done here is we've essentially removed, used solvent to make the brain transparent. We lit up all of the T cells, and so every dot is a T cell, and we can image the entire brain on a massive microscope all at once. And so you can generate a 3D kind of view of the entire brain. And so they're definitely in there. And like the chicken, and so now I'm showing you the density of these cells across all the brain regions. Like the chicken, the T cells are in a specific place. And this is, the details are not all that important, but it's a similar kind of structure in our brain as it is in the chicken brain. And this is the same in humans. And so what we did here was when folks died in the hospital and agreed to donate their bodies to science, and it wasn't a brain injury or they had brain disease, so they were otherwise, their brain should be otherwise normal clinically, we would get their autopsy samples within a few hours after they passed. And we could find, basically using the mouse as kind of a wayfinder, now find T cells in the same parts of the brain that we found in mice. And I remember, I won't forget this because the neuropathologist called me and they were like, "Oh, I guess they're there." The brain is a big place, and there's not many of these cells, but here they are. And I think the most striking example and the most relevant, I think, for food allergy, and we're just starting to get into this now, is the cells that are in the brain from the immune system come from the gut. And so what we could do is we could do these very interesting experiments where if we fed them different kind of diet, we could change the number and the quality of cells that end up going to the brain. And that's basically what I'm showing here. I won't go through the granular details here. And so what we discovered essentially was that the immune system is one of the few organs that can go everywhere in the body. It's a mobile organ. And so it's very good at collecting information at places it's been. They can be encoded in the cell, and then the cell can travel places as essentially cellular messengers of the information of the tissues they come from and transmit them to other tissues, like in our case, the brain. But this could be true generally. And given all of the associations with immune diseases and our moods, our stress levels, um, uh, and kind of the strong association with what we eat, microbiome, um, it seems likely to me that that different kinds of immune cells that find themselves in the brain may actually contribute to not only our health, which is what we show in this study, because we only studied healthy animals, but could contribute to disease. And so this is an area where we're putting a lot of effort into as well. All right, so I'll wrap up here so that we have some time for discussion. Um, my thought is that we really don't understand many of the things, and many of the things we thought we understood turn out to be not totally correct. And I give you a few examples of this. And I think this is important, in particular when it comes to food, because at the moment there's really no systematic kind of study of food as a discipline, in the same way that we have a periodic table so that we can study matter. We don't have this for food. And combined with large gaps in just how the body works and what we thought how it worked may not be totally correct, it makes it hard to understand then food allergy. And I'm I hope I convinced you that the context in which food is consumed seems to matter. So if there's pain and damage at the skin, for example, or if the food contains poisons in it or chemicals in it that have adjuvant properties, or your neuronal state, your mood, and your the activity of your brain seems to matter. And we have, I think, just the tip of the iceberg here in terms of truly understanding all of the chemicals that have adjuvant properties that could make the immune system allergy prone to food. And this is work, all of this work is work that would not be possible without the support of Fawzi, and that Fawzi is very interested in pursuing. And I now kind of speaking as a physician, a lot of modern healthcare is kind of sliced and diced into your heart doctor, your lung doctor, your allergy doctor, and we really need to get back into the patient as a whole, and this includes how they feel about things, their mood, their psychiatric state, and so forth. I hated as an internist to get this these consults from psychiatry to rule out organic causes. Because just because we don't understand exactly how the brain works doesn't mean it's not organic. It still uses carbon-based biology like the rest of the other organic things. And so trying to understand how everything is connected, I think is also going to be really, really important. And so with that, I do want to show a picture of my amazing group, who are really the engines and the creative force behind everything that I showed you. I'm just the salesman here, and all of the various foundations that support this work. I want to, in particular, thank my mentorship, Ruslan, who really transformed the way I think about science and inspired me to tackle very hard and fundamental questions. And I always like to put up this painting, for those of you who haven't seen it. I think it's probably the most important role of a physician, which is the great privilege of being a witness and a participant in someone's life in a most unusual and intimate way, and that our first priority to all of our patients is not to harm them. And then finally, the engine behind me is my family, and so this is us at Easter a couple years ago with the lamb. And with that, I'm very happy to take questions. And thank you for being such a great audience.
Thank you, Andrew, for what is an incredible presentation tonight. I am hoping there's a lot of information here. I mean, two words kept coming to mind: amazing, fascinating. I didn't realize that. That makes sense. So many thoughts that have come into my head. And I'm seeing perhaps we have some in the chat, Caroline.
Yeah, there's one question in the chat. Um,
I asked a question, so I can. It's maybe a naive question, but regarding the stress factor of an allergic response, I hadn't seen that data before. Is it thought to be the cortisol that is I don't know what the word is, like the reason for the anaphylactic response or the allergic response, or is it something else related to stress, or is it unknown? And is that why, like the definition of stress is, I guess, what I'm trying to ask about. And is that exercise, for example, and is the body see exercise as stress, therefore exercise induced anaphylaxis, which we've seen? So just if you could expand on that, I'd be fascinated.
Yeah, I mean, the short answer would be we don't totally know. It's very unlikely to be cortisol because, as you know, when there's some anaphylactic response, it's usually epi, steroids, histamine. I mean, right? I mean, it's part of the cocktail that we give to suppress allergic inflammation. So it's almost certainly not cortisol. Um, the nervous system communicates to the body using a lot of different neurotransmitters. So this is in addition to catechols. So right, there's serotonin, dopamine. There's a lot of these other things that nerves make to make what you think into what happens in your body. And there is a lot of interest in understanding exactly what neuron releases what that may be next to a mast cell. So for example, mast cells turn out to live in your body. They turn out to live basically right next to nerves. And so it's very likely that that neurons discharging certain sets of neurotransmitters can actually activate mast cells and degranulate them. And the question of what is stress, I think, is a very good one. The way I usually like to answer this question is, I think it's different. I'll give an example. If you stayed up all night because you wanted to find out what was happening in the next Harry Potter book, or you were up all night because you were taking care of your baby, it's very different than being up all night because you were cramming for a test or you were anxious about right. So I think exercise is complicated because it depends on the affective state when one is doing the exercise. I think the other thing I'll add there to the sensory neuron aspect of this is, as you know, in OIT, you can't do anything that raises the body temperature. This includes exercise, but hot showers, being in a warm place 2 hours before or after you take the allergen, right? And so there's something. So temperature is sensed by sensory neurons also, and so I think there is some fundamental connection here that we just don't totally understand. That our group and many other groups within and without the Fuzzy Consortium are actively trying to understand using animal models and in humans. It's a great question. It's something we all have experienced and as clinicians observe, but don't have a good explanation for.
Thank you.
Andrew, when we spoke a while back, you had mentioned something along those lines, what you just were explaining, and mice when they were, and I may be remembering this incorrectly, but if they were experiencing anaphylaxis, they would try to find a cold place to go.
Yes. I was cognizant of the time I had, and so I didn't want to cram too much in this. But yes,
I found that fascinating. I think it's worth sharing.
So yeah, a lot of times when folks and scientists do experiments, mice are kept in cages. They don't have a choice at what temperature they want to be in. They don't have a choice of what they want to eat. They don't have any choice, unlike us, and unlike mice in the wild. And so, because of this observation of not doing anything to increase body temperature in OIT, and also many observations of, like, if you get stung by a bee out camping and you left your EpiPen somewhere, you should jump into a cold lake. These kinds of anecdotes. We did a very simple experiment, which was if you anaphylax a mouse and it had a choice on which temperature it could go to. Mice ended up voluntarily finding otherwise cold places that they would normally not want to go to, that they normally would find actually quite aversive because they're small animals and small animals don't like the cold. And when they found the colder place, they were also able to survive anaphylaxis better. And so we this study also is we're currently revising, and we're actually gearing up to test whether or not if you applied cooling creams in children getting OIT, would they actually have less GI distress and adverse events? Because the main limitation with OIT is the dropout rate. If folks could actually stick with it, it's actually not bad. It's just very hard to tolerate. And so is it possible that something as simple as tickling this sensory cue could make it more tolerable, and then we could kind of make the immune system more tolerant to peanut, for example? But it's these kind of fundamental questions, Jen, I think that are missing from the body of knowledge that I think would be very, very helpful in coming up with, in this case, it's I would say a pretty practical and relatively low cost potential solution to something that is otherwise a barrier.
Absolutely, absolutely. And I feel like it's just right there, ready to be accessed with the work that you're doing. We are after 8 o'clock. We could take one more question if there is any, but I don't believe there is. So let me just say this. I would like to again thank you, Andrew, for sharing such an incredible wealth of information and helping us understand the research that you and your colleagues are doing to help us better understand food allergies. Again, even just hearing about immune cells in the brain, a new discovery, it's all fascinating to me. It's beyond my capacity to do it myself, but to learn about what you're doing is just fascinating because it is clearly bringing us closer to understanding food allergies and to a cure and to the whole body. I love that Fozzie and you and your team and the other teams are looking at this from the whole body. And that's, I think, going to bring us so much more valuable and bring us closer and closer to developing a cure. And so I guess my final message here tonight is we all learned so much. My guess is we'll all have questions later. But this is progress. What's happening in Andrew's labs and others, this is progress for all of us impacted by food allergies, and this brings us hope for a world without food allergies. So again, we thank you, thank you, thank you. If there are any questions, it was an excellent information. I agree with you, Karen. As always, if you have any questions about food allergies yourself, please encourage you to contact your healthcare provider. Preferably, you have a board certified allergist or someone that you're working with that can help you work out some questions that you might have. Before we close tonight, I want to share that if you don't currently receive AFA New England's emails and the important notifications that we send, or our asthma and allergy bulletins that we publish 3 times annually, we do encourage you to join our community so that you receive these notifications, so that when we plan our upcoming 2026 Speaker Series webinars, you'll hear about them. And when we do our winter bulletin, you receive it. To sign up, it is simple. Go to our website, asthmaandallergies.org. Long, all connected, but easy. Asthmaandallergies.org, and that will bring you to our community, and would love to have you welcomed if you're not already a part of it. So again, that brings us to the close of a phenomenal evening. And we thank you all for joining us. And again, Andrew, thank you for all that you do and for presenting with us tonight.
It was my
We
great.
hope you all have a good holiday season and enjoy. Thank you all. Good night.
Good night.